This presentation actually it's pretty short and very specific. Essentially, I will report the results of a single scientific publication. The reason why I'm presenting it because it's actually a fruit of Fin series flagship. It would not have been feasible without it. It's a fundamental finding, a serendipitous one, and it's a collaboration. Collaboration between Tekla from VTT and then Yoshi Nishiyama from Grenoble. And Zhujun Meng was a postdoc, joint postdoc for me and Tekla, who worked exclusively on the flagship research. Now, the motivation behind the research I'm presenting was to explore the condition under which dissolved xylan can be crystallized into nanocrystals, which is fairly unusual, and to comprehensively characterize the emerging xylan nanocrystals thereof. Now, I'll give you first some background on polysaccharide nanocrystals. Many of you might be familiar with that. I'll go through the experimental setup, how we can come up with the xylan nanocrystals, and then just explore the characteristics, dimensions, crystalline structure, and how the growth of the crystals can be controlled. Now, these are familiar figures, especially the one on the left here, like cellulose nanocrystals. They have a rod-like morphology. They have been, let's say, en vogue since Derek Gray found out that they spontaneously form chiral nematic liquid crystals in the early 1990s. You can extract nanocrystals also, for example, from starch. They have a platelet-like morphology. They are not as fashionable maybe as cellulose nanocrystals, but they are there. And the way you prepare the nanocrystals is top-down, like virtually all nanocellulose species. Not every single one, but so you start from fibers. In fibers you have microfibrils. They consist of microfibrils, and essentially nanocrystals are cut microfibrils, and the cutting is done by a means of harsh acid hydrolysis under a fairly narrow reaction window. 64% sulfuric acid, aqueous sulfuric acid, 45 degrees, somewhat elevated temperatures. It can vary a little bit on the species of plants you're using for that. Anyhow, it's a top-down route. The same with starch nanocrystals. You prepare them usually with HCl hydrolysis. Now, hemicellulose is a different species altogether. I mean, they form mostly amorphous structures, as you may know. So that's what you learn from plant anatomy or wood anatomy during the first courses, if you take them at the university, that there are crystalline cellulose microfibrils, highly crystalline cellulose microfibrils, which are embedded in amorphous matrix of hemicellulose and lignin. Now, if you isolate hemicellulose, it can, there are hints at crystallinity if you do X-ray diffraction, but only hints. You usually get something like 10% crystallinity index. So if you subject hemicellulose to a similar acid hydrolysis as you would do with the cellulose nanocrystals, you get pretty much monomers or their byproducts. If you want to get crystals out of hemicellulose, a bottom-up route of dissolved hemicellulose would seem like the right thing to do. Now, so we set out here with a highly straight chain xylan extracted from birch. There's a publication quoted where, if you want to actually get into the details of characteristics of that hemicellulose, there's very little other stuff than xylan. So it's pretty much straight chain xylan with methyl glucuronic acids dotted here and there, maybe 1 in 20 units or so. So a straight chain, we were not actually trying to come up with xylan nanocrystals. This is something we don't say in the publication, but I can tell you here, it was a serendipitous finding. We were trying to dissolve xylan in order to have ultra-thin films out of it. And you may know that hemicellulose is notoriously difficult to dissolve in practice, although DMSO is usually seen as a good solvent for hemicellulose or for xylan as well. And so Zhujun, our postdoc, she was trying to dissolve xylan, and of course, when it doesn't dissolve properly, what do? You start heating it, and then you wait. And she started noticing that there's some kind of precipitate after waiting for a while at elevated temperatures, and she took a look at what the precipitate was with a microscope, and they were these nanocrystals, longitudinal nanocrystals, not entirely different from cellulose nanocrystals. So all of this happened under slightly elevated temperatures at dimethyl sulfoxide solvent. Now, this is really a bit more in detail picture what the dimensions look like. So the crystals I would say they are fairly long, hundreds of nanometers with a peak at 900, 1000, or around micron long, but a clear distribution in the length. There is a cross section you see which is not a square. The cross section is actually it shows that it is slightly platelet-like. So it's around 10 nanometers. They are around 10 nanometers high. They are like flat platelets. These are AFM images. The width here of the cross section is obviously exaggerated, but it is still considerably wider than it is high. So it is not like a cellulose nanocrystal in that respect that it would be like a stick, but it's more like a platelet. The molecular weight here, the average DP we have here is a bit over 100. So the xylan, as most hemicellulose, is relatively short chain. And we started to look at a little bit more what these look like. I mean, what they consist of, these nanocrystals, and we did obviously solicited NMR. We did a very simple thing, and we noticed the DMSO peak there, and then we did TGA, so thermogravimetric analysis, and we saw a peak at 189 degrees, which is the boiling point of DMSO. So there's obviously DMSO plays a role in the structure, right? And when we put it in water, the nanocrystals, the morphology totally collapsed. So it was destroyed. So the presence of DMSO is crucial in stabilizing the crystal structure. So water obviously dissolves DMSO. It's a pretty polar substance, right? And then we went to Grenoble and wanted to see what the crystal really looks like, because we did not have a proper means to really crack it out, have a crack at it ourselves. Here's actually on the left hand side, there's a very nice TEM image of a platelet-like crystallite. We did electron diffraction and X-ray diffraction. They did in Grenoble, and what we noticed that the xylan chains, they are aligned along the thickness direction of the nanocrystals. So it's like a longitudinal slab, right? And they are aligned on the thickness level. So they are perpendicular against the longitudinal direction, the chains, and they fold a bit like a polyethylene crystal, right? And this was rather surprising. They also packed in an anti-parallel manner in a monoclinic unit cell. So that we have quite a nice crystalline structure, and the DMSO is inside the crystallite. That's also something I have to emphasize. Yeah, and you see maybe the DMSO is here. Here is a better picture of how the DMSO is incorporated in the crystal, and it is essential for the crystallite. So I emphasize, if you put it in water, the whole morphology collapses. The crystallite is destroyed because the water dissolves the DMSO. And these crystals are tunable just with time. We tried different temperatures and they didn't work very well. That's the lower images here. So this 60 degrees that we originally tried and it was a lucky strike. So the crystallites really form under that temperature, and you can control the length with the growth time. The growth time is it's days. You see, it is the 2 days is the shortest span here, and then up to 20 days or even more. The dimensions are almost monodisperse, and they are monotonically increasing with time. So to conclude, the xylan nanocrystals can be constructed bottom up from dimethyl sulfoxide solution under elevated temperature at 60 degrees specifically. The xylan is, as you may know, a twofold helix in solution, but it's a threefold helix in crystal, and the DMSO is an integral part of the structure, the DMSO molecules of the crystallized structure. And importantly, the change aligned in the thickness direction with the towards the nanocrystal, and it's feasible with chain folding. So it really makes this kind of structure. And the length of the nanocrystals, it could be tuned with time. And guess who I want to acknowledge for funding, and then I would finally like to acknowledge you for the attention paid. Thank you.